Okay, the first thing we're going to talk about today is the use of a microscope, and then we're going to go through the parts of it and how to focus it. The first thing you want to know is how to carry a microscope. Whenever you are going to carry a microscope, you need to make sure you're holding one hand on the arm of it and one hand on the base of it. Then you need to make sure you hold it up close to your body. That way it doesn't it has a less likely chance of ending up on the ground. Alright, the next thing you're going to want to do is take the cord, which in some labs will probably be wrapped around the base of it, and that should just keep accidents from happening. Then plug it in, and when you're unraveling the cord, just unravel enough in order to plug it up. Don't unravel all of it, and then again that'll keep accidents from happening. Okay, once you do that, you're going to turn the microscope on. The on-off switch is actually located back here on this microscope by the plug-in. Now, another thing, if you notice, I'm not sliding the microscope back and forth across the table. I pick it up. The best thing for you to do once you get started is, if you need to look at something, don't slide the microscope back and forth across the table. You can mess up the objectives. You can also mess up the mirrors inside the microscope. So the best thing is for you to move and leave the microscope where it's at. Okay, the first part of the microscope we're going to look at is the eyepieces. Now, this is called a binocular microscope because it has two of these. These are also called oculars. They rest on what we call the headpiece, which is this part, and it actually does rotate back and forth. Then, of course, we mentioned the arm earlier. If you look under the headpiece, you'll see these four rotating objective lenses, and they are all different sizes. You will need to know each of the magnifications for each of the objectives. For the shortest one with the red line, that's the scanning objective, and it has a magnification of 4x. For the next objective, this is low power, and it has a magnification of 10x by itself. The next one is the high power with the blue line. It has a magnification of 40x. And then this last one, which you won't use in anatomy and physiology lab, it's actually called the oil immersion lens or objective, and it has a magnification of 100x. Alright, right up under the objective lenses, you'll see the black platform. That's called the stage. And you'll notice that I have a microscope slide in here. In order to place your microscope slide and secure it, you're going to take your microscope, and using this little piece up here called a stage clip, you're going to pull it back with your finger, and you're going to slide the microscope slide into place. Okay, don't just let this piece go. Ease it back so that it doesn't break your slide. In order to move your slide, I'll turn it this way. In order to move your slide left and right or backward and forward, you will use the knobs up under the stage. One moves it backwards and forwards, and the other moves it left and right. Okay, underneath the stage, you'll see this piece right here. It's called the condenser, and there's another little lever right here. It's called the iris diaphragm. It allows you to regulate the amount of light that actually enters the microscope. Okay, then below you have the illuminator. That's just basically where your light comes from to go up through and light your specimen. On the base, which is this piece, you see a red knob. That's the light control, and it's just going to control the dimness and the brightness of the light. Now, in order to move your stage up and down and to focus your microscope, you're going to use what we call the coarse adjustment knob and the fine adjustment knob. Okay, the coarse adjustment knob moves it a whole lot at a time, and then fine adjustment or fine focus moves it just a tiny bit at a time. Now, to focus your microscope slide, you're going to want to make sure that whatever your specimen is that you're looking at is about center of this light source right here, and that your objective, your smallest one, is clicked into place. Next, you're going to take your coarse adjustment knob, and without even looking into your microscope, rule of thumb is to go ahead and roll your stage all the way up until it stops, and you'll feel it stop. Once it stops, then you're going to look into your oculars, and you're going to locate your specimen. You will do that by rotating the coarse adjustment knob again back towards you. Once it is focused, then you can take your objective and click the next one into place, which is low power. Then looking back into your microscope, you can use the coarse adjustment knob again, and then rotating it away from you until your specimen comes into your field of view, which is what you're looking in the microscope. You're going to use a fine focus to make sure that it's completely as clear as it's going to be, and then you can rotate to the high objective. Now if you'll notice right here, you'll see that your slide and your objective are very, very close together. That distance is called the working distance between your objective and your slide. And since this is so close right here, you do not want to use the coarse adjustment knob on high. You only want to use the fine focus, because if you were to use the coarse adjustment, it could break your slide. Once you get on high power, you shouldn't have to rotate the fine adjustment very much before your specimen comes into view. It may also help you to increase the brightness of your light to help you see it a little bit better. Now, in this class, like I said, you won't use that fourth objective, the oil immersion. That's going to be more to view bacteria and very small things like that. Now, once you're finished viewing your specimen, then you would want to rotate backwards to your smallest objective. You're going to want to make sure you take your slide out using your mechanical stage, your stage clip, and then you're going to roll your stage all the way back down until it stops. You're going to dim your light and then turn your microscope back off. Then you want to unplug it and wrap the cord back around the base of the scope and then secure it to make sure that, again, accidents don't happen. Make sure that you clean any glass with lens paper before putting your microscope up, and again, make sure that this is locked into place. Another thing to check is over here, when you were using your mechanical stage to move your slide back and forth and up and down, it moves this piece right here. So you want to make sure that you get that back over to where it's almost centered, so that no pieces are sticking out so that they don't get broken. Then once you've finished all of that, then you want to put your cover back on your microscope and place it back in the cabinet where it belongs.